up since 2017. We are organizing offline events, maybe mainly, and also organizing online events. We're also happy to see all of you today. So, like both allegorically and literally. So if you could turn on your camera, it will be nice for us to see each other, if you feel comfortable with this, of course. So talking about 3 years in Munich, we had quite a lot of offline events, but currently we are blocked on this because of obvious reasons. Coronavirus is hitting everyone. And so we are trying and doing this new format of chit-chat sessions. So this idea of this is there is one speaker, one topic, and there is an opportunity for everyone to ask us many questions as he or she wants and communicate directly with the speakers. Something which is unfortunately not that easy to do on offline events. So today we are having Max as our speaker and our guest. So Max, say hello. Max has background in pharmacology, but currently he's doing PhD in field of microbiota in immunology in Israel, and he's going to talk about diagnosing infections. And this few words about us, then we're going for the talk, and then we are going to have the question sessions when everyone had an opportunity to ask us many questions as he or she wants. And finally, we're going to have a networking session when we will have a chance to just chat about random stuff. And so without any delays, let's go for the presentation from the Max. So I'm sort of sharing the screen. So Max, the stage is yours. Please go on.
Yeah, 1 second. Is it all right? Yes?
Yes.
Yes, okay. So, yes, thank you, Evgeny. Today we're going to be talking about diagnosing an infection. And first of all, we need to remember that our bodies are constantly immersed in a world full of various types of microorganisms. There are bacteria, viruses, and so on, a lot of them. And the situation when some of them are messing with us, messing with our physiology, and make us sick, this is the situation we are calling an infection. Today we are going to talk about the first step of treating almost any infection, namely diagnosing an infection. At first, let's dive deeper at the question, what is exactly an infection? So our body, its inner environment, both liquid state, inner environment like blood, or solid state like cells, muscle cells, fat cells, and so on, they are meant to be sterile. So by default, there mustn't be any microorganisms, outside microorganisms inside. So in order to achieve this, our body forms some thick or maybe thin layers of cells tightly bound together, like skin cells, cells of our gut epithelium, and so on, which are meant to protect us from the microorganisms which are lingering outside. Those organisms, they are generally not harmful to us. So they are just lingering there. They can be even beneficial to us. But sometimes something may go wrong. Sometimes some of those basically friendly microorganisms can become hostile, or some outside microorganisms can come and they can destroy this layer of protection. They can penetrate the body. And then both them, bad microorganisms, and also some good microorganisms can get inside, so that our inner environment become non-sterile. We become infected. So infection is some harmful microorganism inside our body, or some microorganisms in the wrong place, like blood, brain, kidney, and so on. So places which are meant to be sterile. And diagnosing an infection is finding out which organism caused this infection, where is it located, and sometimes we can even find out with what we can treat it. The goal for diagnosing is picking the right treatment. So how do we do it? How do we diagnose? There are basically four methods. The first one is diagnosing by symptoms. It's the oldest one, and it's based on the notion that each and every disease out there has some symptoms, some like changes in our behavior, in our physiology, in our feelings related to this disease. Infectious diseases also have those changes, but there is a problem that those changes are almost common to all infections out there, no matter which organism caused them. This is caused caused by the thing that, as well as microorganism, is like messing with us, with our body. It produces some poison, some chemicals, and it causes what we call specific symptoms. But also, this microorganism induces our immune system, and our immune system also changes the physiology of our body, causing what we call non-specific symptoms. And most of the time, when we are looking at this, in clinical context, we see that those non-specific symptoms vastly overshadow the specific symptoms the microorganism can cause inside us. So what are those symptoms? The non-specific ones, we are all common with them. They are some things like cough, edema, redness in the place where the infection came in, and so on. So those are what we call inflammation. Inflammation is a common response to any infection, almost any infection in our body. And specific symptoms are something like changing the color. So for example, if we see something green on the green screen, green nails, something like this, we will know which organism could cause this. Some smells, specific smells to some specific infections. Some localization. Localization, I mean some organs, like our inner environment is quite harsh. Like not any living being can live inside us. And so if we see an infection of the brain, there are just a handful of bacteria or viruses which can survive there. And this way we narrow the search. Like if there is a brain infected, then we can know, okay, there are like 10 species we can look for. Behavioral changes also. The most common example here is rabies. During rabies, the patients experience aquaphobia. They start to be afraid of water, and they experience also they got afraid of light, bright light. And there are some observations we can also make. For example, if we find bacteria in the urine, also not any bacteria can live in the kidney. So if we see bacteria in the urine, we will know, okay, we can narrow down our search. If there are some specific symptoms, it's the best way to diagnose, because those symptoms, it's cheap. It's almost free to look at them. And we can do it like in a glitch, like in seconds. So these are some symptoms. For example, here we can see bacteria, those violet dots in the blood. And here we can see green nails, like changing the color of the nails. This can be caused by only one bacteria called 𝗤𝗿𝗄𝗎 aeruginosa, and we know that we need to treat it. Like this is it. This is 𝗤𝗿𝗄𝗎. We need to take specific treatment for it. The second method we can use is cultivating. It's a very old method, a bit newer than symptoms, but also very old. It was developed by Pasteur back in 19th century. And what he basically did, he took diseased animals and took all the samples from them, like samples containing bacteria, put them on the plate, and let them grow into colonies. Like if we take bacteria in the good environment, they will start growing, spreading, and form a visible colony. From that notion, we went a long way, and right now, for various conditions, for both healthy and diseased individuals, we have databases which bacteria can form which colony. There are databases explaining all kinds of like properties of these colonies, how fast they grow, which size they can achieve in which conditions, like how do they look like, what are their colors, and so on, how do they stain with different chemicals, and so on. And by using this method, we can cheaply diagnose an infection. This is how it looks like. We took a sample from a person, plated it, and this is what grew. So as you can see, those colonies are various in shape and sizes and colors. So if we have this database, we can distinguish each of them and find out if it's harmful or harmless. So the good thing about cultivating is that it can detect only live organisms, so we won't be mistaken by trying to combat something which is already dead. It is very old and established method, so there is a lot of data about it. It's very simple and cheap, and you can do it in remote areas, and you can easily screen a lot of people for some disease. The problems are that it can be very time intensive. So for some bacteria, it would be hours. For others, it would be weeks or even months. For example, tuberculosis bacilla can grow up to 6 weeks before you can actually see the colony. You can't detect viruses by this way. We can cultivate viruses, but not for diagnostic tasks. You can't detect unculturable bacteria. It happens to be that almost all bacteria in the gut, for example, or on the skin can grow on the in the media on the plates. We just don't know the conditions they will grow on. So this is it. This is called cultivating. The next method would be PCR. It's already a modern method. It was developed in 20th century, and it's based on the notion that each virus or bacteria contains the thing we call genome. Genome is basically an instruction of how to build this organism from scratch. It contains information about unique information about this particular virus or bacteria. And what can we do? We can design the things which we call primers, and if they are chosen right for this particular species, they will stick to this genome under certain condition and initiate the reaction we call PCR, the multiplication reaction. They will start to copy the region between them. And this copying is so immense, the amount of copies so large, then that we can actually see this DNA, this amplified part of the genome, with our bare eyes. If we are mistaken, for example, took the primers for wrong kind of microorganism, nothing will happen. They won't stick to it, and the reaction won't occur. So in order to visualize this, we use the method which is called electrophoresis, which is this is how it looks like. It's almost one-to-one photography. So here we can see that samples 1, 6, and 7 has those amplified parts of the genome. They are of defined length. So we know that we are looking for this. It's not some kind of mistake. And samples 2, 3, 4, 5, and 8 don't have anything. So the persons 1, 6, and 7 were infected, and 2, 3, 4, 5, and 8 were not. So what's good and bad parts of PCR diagnostics? The good thing is that it can be very, very specific. It's very time effective. No matter the agent, the infectious agent, it will take only 1 to 2 hours to detect it. It is very easily automatable, so we can use robots to do large scale production. The reagents are quite cheap. And we can detect even before the symptoms. So like a person just got infected by COVID infection, in several hours, we already can detect this virus in the blood or in saliva of this particular person. He doesn't have any symptoms, but we can detect it. The problems are that we need initial knowledge, so we can't just do it from scratch. If some novel pathogen arises, we need to know the sequence of its genome, like what's the genome composed of. you require some personal and specific tech, so it's not something you can do in remote areas. And there are some problems which causes some samples to be false positives. For example, if you are not working clean enough, the even small contamination of one sample from another sample can cause the negative, basically negative sample to be positive. And if a person have recovered, the genome is almost the first thing to be degraded. So if a virus or bacteria is dead, it won't contain a readable genome probably. It can contain, but the genome will degrade very fast. And the fourth method I want to talk to you about is antibodies. Antibodies are large protein molecules which our immune system develops in response to the viruses or bacteria or some other infectious agents. At first, then when we first meet this agent, there are no antibodies against it, but in 7 days, a normal time for almost any infection, the body produces those molecules and they specifically bind to this virus or bacteria. This region of the antibody is called the variable region. It can adjust to almost any virus or bacteria out there. And in our body, in our blood, those antibodies, they remain for months or even years, and sometimes they remain for the whole life. So we can detect them there. How do we do that? We take some plates, we glue our virus somehow to the bottom of it, and then we add the plasma from this particular human, where the blood plasma from some human we want to check. Then we wash this plasma away, and then if there was some antibodies there, they will stick to this virus. And after that, we add some reagents which can show, are there some antibodies inside this well or not. Here you can see how it looks like. The blue wells are the ones positive, and the white wells are the ones which don't contain antibodies after washing. So like here we can test up to 96 people at once. The good things about it that it can be also very specific. It's also very time effective, and it also can be done by robots. And the idea is that it can detect those who already recovered. We can find people who, for example, were infected with coronavirus, novel coronavirus, but didn't got any symptoms and recovered on their own, and we can find those people and, for example, use this data for contact tracing or something like this. The problem is that it's much more expensive than PCR. It can't detect the infection right after it occurred. You need at least a week to develop those antibodies. It's also prone to false positives and also false negatives. For example, sometimes we can't find any antibodies if we're glued not a right part of the virus, for example. So these are basically four methods: observations of symptoms and culturing methods, PCR, and antibodies. So when you are reading next time, for example, some, I don't know, news article about coronavirus testing, you will know what they basically are talking about. So this is it. I'm open to your questions.
Thank you, Max, for the interesting talk. So now we'll go for the question part. Before this, let me just say a few words about how we would like to do it. Could you stop sharing the screen, Max? Oh, thank you. Yes, so please turn off your microphone, which is already done, and turn on your camera if you want to ask the questions. And also raise a hand here inside of the Zoom in order to do it. So to do it, you need to click on this icon. And then in the chat at the right, you will see, in the list of participants at the right, you will see this icon. And so please raise your hand, and then you will get the opportunity to ask a question. You also can ask a question in the chat if you want. So just type your question there. Just open the chat and type a question. And also for the people who are following our streams on Facebook and YouTube, you also can ask your questions there, and then they will be reposted here. And we also will try to ask them. So the first question, I want to use the opportunity as a moderator to ask a question which interests me personally. So you mentioned COVID. So how exactly you will use these methods to test for the COVID? And also more precisely, did I understand it correctly? For example, this PCR, until you know the genome of the virus, you cannot test for this?
Yes. For coronavirus, modern coronavirus, we can use only two of those methods. Those are PCR and antibodies. So PCR we can use for initial diagnosis when we first, like a person encountered someone infected and just wants to know, or doctors wants to know if he got infected or it didn't stick to him. So we will test this person's saliva with PCR over and over again, and if the response would be positive, then we will know, yes, he got infected. If it would be negative, then no. And with antibodies, we can test if this person didn't develop the COVID. Symptoms for COVID also can be helpful. For example, there is a specific symptoms only for this disease. It's loss of smell. So it's the most common one. And culturing methods are not applicable. So this is virus, it's not a bacteria. We can grow it for scientific purposes, but it's very expensive and hard to culture viruses, so you can't diagnose with this.
Okay, thank you. Lawrence has a question. So could you please turn on your camera and the microphone?
Hi, yeah, I have a question regarding Corona as well, kind of. So actually the first question is, what does it depend on how long this antibodies stay in your body or in the blood? Apparently it kind of, it depends on the person as well, because from studies that I read at least, like some 40% or something are not, do not have this antibodies anymore after a while. So what's the reason for that?
The thing is that our immune system possesses several means to combat infections, and antibodies, they are one of the common ones. So most infections are combated with antibodies in our immune system, and it's the easiest to test. Like for almost 100 years of diagnostics, people just got used to using antibodies as a method of diagnosis. So it's the first thing we'll look for. But with COVID, it seems that other branches of immune system are much more involved in the fighting with this disease than antibodies. So our body just don't develop antibodies, good antibodies against this virus. So it seems that we will have to use some other methods. So antibodies, they are developed, but they are not long. They are not lasting long. In some, they stay. In other persons, they stay for like several weeks and then they go away.
But there is a theoretically some method how you can check if person had the COVID before, apart from antibodies method?
Yes, there are. So the antibodies is not the only adjustable molecule in our body. The other ones, the other branches also have means to remember the stuff. Like antibodies is a way to remember the infectious agent. Like if they are developed, then immune system just remembers this virus for the future. If you got infected with this virus once again, the antibodies are already there. They will cover the virus, it can't enter the cells, and the antibodies are basically like lighthouses for the immune system. They signal that I've caught something bad, come here and did it. So there are other molecules. They are basically tied to the cells. They are not free-floating like antibodies. They are tied to the immune cells. They also work almost like antibodies. So yes, we can test on them, but right now not routinely. It's not something you can do in any lab. And probably you will be able to do it in any lab after these pandemics, because like it showed us that we need to develop such methods.
But I'm not a specialist on this. I think why people are concerned with antibodies is also because there's an opinion which is, if you have antibodies, you cannot be reinfected. But if you don't have, you can be. So is it true or not?
There are several things why people are concerned with antibodies. First of all, yes, if you have strong antibody response, then it is much less likely you will catch this disease once more. Any disease starts with like several dozens of viral particles get in your body. And if there are a lot of antibodies there, they will just be covered with them, and they will be harmless. They will be eaten, they will be destroyed by the body. The second thing why people use antibodies is contact tracing. So if there is some community where people got infected and we, for example, want to find out the source, we can test the whole community or many people in there for antibodies and find out who got infected, who he was contacting to, and so we can build a map of spreading of the virus, even if some persons didn't develop any symptoms. And the third is a very important thing. We can use the blood of those people who recovered to cure the ones who got the disease recently. It's called passive immunization. So if a person developed COVID and got rid of it, got like healthy, and has a strong antibody response, then we can transfer those antibodies to the diseased person, basically transfer the blood to transfer this protection. It won't be lifelong. Those antibodies will stay in the blood for like several weeks, but they can treat an acute infection in the susceptible individual, for example. This is how we treat rabies. Like rabies, you can be immunized from rabies and you can have your own immunity. But if you didn't get immunized and you got, for example, bitten by a mad dog, and you go to the doctor, I was bitten by a mad dog, I'm afraid of rabies. Doctor will say, okay, I got a horse which was immunized by rabies, and it produced so much antibodies, so I'll basically give them to you right now. No matter those are horse antibodies, they will still work. And if you got rabies from this bite, you will be treated.
But how? Interesting. I never heard about this technology. So how technically it's done? It's a blood transfusion, or it's somehow like a liquid which is produced like a.
No, it's not blood transfusion. Like if you're in some bad situation when you don't have access to anything, then yes, if you have one person who was immunized against rabies and the one who got bitten, you can just simply make a blood transfusion if their blood groups are matching. But the most, like how it's done right now, we take animals, big, large animals, we immunize them with this specific virus or bacteria. We get blood from them. We purify antibodies out of it. So we get only the antibodies, not all the other proteins that are out there, not the cells, not proteins, just antibodies. And the solution of those concentrated antibodies can be used as an acute response. Like we can use it right now to save this person. Because if we give him a normal vaccine right now, it won't probably have enough time to develop antibodies until the rabies spread inside him. Sometimes it works, sometimes it does not. Having those antibodies is just a good idea. We have them for tetanus, for example. We have them for rabies, or something like this.
Yeah. Okay. Thank you. Thank you, Lawrence. Someone on your video want to ask a question, if I understand correctly.
Yeah, I'm Carol. Nice to meet you guys. Because you mentioned that we immunize animals to culture antibodies. Which animals are we using at the moment, actually, besides horses?
Horses, camels, some big animals. But it's a very old technique. So right now we can do it a different way. We can just take the cells producing from antibodies from a human, actually. So this is like an ancient tech. It's still used in some areas, but right now we can just do human antibodies if we want to. It's just a bit more complicated and a bit less funny than taking a horse.
But does it mean that you need to have a person who volunteer to give the blood essentially for this?
You can have either person, or you can have a mouse with human immune system. There are such mice.
Oh.
They develop human antibodies, so we can take those mice with human immune system. We can immunize them. Then from them we need to extract just one cell which produces those antibodies. Then we do this cell cancerous, like we intentionally do it cancerous. It starts to spread and produce antibodies in the media. So we basically grow it in the biomass, which just produces antibodies. And then we can culture it for years and just extract antibodies and use them as a treatment.
But can it work with the coronavirus, for example, this approach?
Probably yes. I don't know if there was some such research. Right now, what they do, they just use recovered individuals to treat the disease plants.
It's interesting why people are not talking about it more, then, because it means anyone who had it can just give some blood, right? And then essentially provide the cure for someone who get it late, right? Or
actually, you probably just didn't heard of it because in Israel here they use this technology. In Russia, I heard they use it. In Britain, I also heard they used it. So it's quite a common thing. Just the problem is that it's not a drug which you can register. It's the thing you can do, but it's the thing you do on the daily basis. It's not like something you can register and say, okay, we have a panacea, we have something which will cure everyone.
So you need to have you need to do it fast, right? So you extract it from the body and of the person, and then you extract the antibodies, and then you inject them in a sick person, right? But you cannot store it.
Normally, you even don't extract the antibodies. You just you take the blood, you separate all the cells. So and all what's left, the plasma, you can use it for transfusion.
Interesting. So very interesting question.
The problem with coronavirus is that it's not coronavirus what kills. So if a person already developed acute symptoms and is basically dying, antibodies against coronavirus won't save him because he's dying not of coronavirus, he's dying of other infections, which coronavirus made possible to become severe.
So what kills exactly then in case of coronavirus?
Other viruses and bacteria which get in the lungs. So coronavirus, what basically it does, it destroys the immune system in this particular area. And all the other stuff which is out there just kills. That's why they say that if you look at the graphs, much more people are infected now than in spring, but much less are dying right now in percentage. Why is that? One explanation is that right now in the northern hemisphere, where all the most of the people live, there isn't a flu season. So there is not so many those respiratory infections right now to kill the persons who got COVID. And if we let the infection spread from this point, then by autumn, when the flu will return, it will find a very suitable environment for it.
So you think it's going to be spiking both in the number of cases and also in lethality?
Not in the number of cases, they are not affected by the season, but in the lethality. So it can be a disaster if this is right. if this idea is right, it can be very problematic.
Yeah, this is kind of worrying perspective. Yeah, I wanted to ask you on slightly different topic. So, could you say a few words about your research on autoimmune diseases? So, is it something which you're studying? Maybe.
Yes, I studied them back in my master's degree, and I probably will continue with that right now. But it's not defined yet. I just got to my PhD course, so we don't establish my thing. So what we tried to do, we tried to cure autoimmune diseases with the way people right now are trying to combat some cancers. So
maybe you can say a few words. What's autoimmune diseases? Because I have very general idea about it.
Okay, autoimmune disease is a type of disease where our immune system recognizes some part of our body as an infectious agent. So it starts working against this kind of this part of body like it was an infection. One example is beta cells of pancreas, which produce insulin. So if those cells are recognized by the immune system as hostile cells, they begin being killed by the immune system, and everything else is fine, but those cells are killed. They don't produce enough insulin, so people got dependent on the insulin injections.
So it's diabetes.
This is diabetes, yes. This is type 1 autoimmune diabetes. So, and there is a method to treat cancer. When we take immune systems from the body, we stimulate them to be like much more aggressive, and then we inject them back in the body, and they kill the cancer. Like normally, cancer cells are meant to be killed by our immune system, and those cells, if they manage to escape this killing, then the cancer develops. So there is a method to teach the cells to escape this suppression by cancer and just inject them back in the body. So what we were trying to do, we were trying to replicate this with autoimmune diseases. In our immune cells, there are killing cells, which are inducing inflammation, kill the cells infected with viruses, kill bacteria, and so on. So they are like sword in our protection. And there are also cells which called T suppressors or T regulators, which do the opposite stuff. They protect the healthy cells from the immune system. So they make sure we don't get autoimmune disease. When autoimmune disease is there, then some of those cells just somehow disappeared or haven't been produced at all. So what we can do, we can just take those cells which are attacking those pancreas, for example, cells, and we can convert them to protecting. Not killing, but protecting. So that's what we were trying to do, and we were even successful up to some point. So right now this work continues back in Russia, where I'm from. So hopefully we will have something.
Do you think it's possible what in our lifetime there will be find a way to cure diabetes?
I think yes, because diabetes. There are two things about diabetes which make me optimistic. The first thing is that it's immensely studied. From immunological point of view, it's a very simple disease. It's one type of cells with one protein attack. So it's like if you want to some simple target, you take diabetes mellitus type 1. So people study it a lot. And the second thing, it's that there are pharmaceutical companies, which made their fortune on producing insulin for diabetic patients. And right now, what we can see that they are like panicking. They are investing in different areas, trying to make more things. Like up to some point, making insulin was a great idea because it's very cheap to produce, and it can people need it the whole life long. So like it's a fortune out of nowhere. So the first ones who got in that train got really rich. And right now what we can see, it's a panic. Like the companies, they just trying to spread, like Saudi Arabia right now. They trying to spread from oil to different kinds of high tech and so on. Also with those pharmaceutical companies, they know only how to produce insulin. They have a lot of money. And right now they are trying to make more stuff, just hire new researchers, buy something, but some startups just to like do a decent pipeline before insulin stops.
But this is very old conspiracy theory, right? What these pharmaceutical companies are actively suppressing the
research
viewers essentially on this because it's fortune. It's like drug selling, like even more profitable than selling drugs.
In modern world, it's quite hard to suppress such things. Because, as I said, diabetes mellitus is a very immensely studied disease. So what they do, they don't suppress the research in this way. They trying to get ahead of this. So they understand that their time has passed. That in several years, 1 or 2 years, 9 or 5 years, we will have a final treatment, because it's a very immensely studied disease. And there are a lot of like prospective studies, like many things work in mice, many things works in the various other models. So they need just several years to get to humans. And those companies, they are trying to get ahead of this wave. They are trying to buy startups, buy patents, just to be the ones who would be the first to distribute the final treatment.
It will be incredible.
Hopefully, yes.
Yeah. So thank you for answering the questions. It was pretty interesting. I got to know a lot of new stuff. Let me say a few final words. So if you enjoyed what we are doing, we are kindly ask you to help us, because currently the situation is COVID is not easy for anyone, and also the companies are not that keen in supporting volunteering work. So our organization is fully volunteering organization. No one is getting paid. We're not getting any profit out of this. We're just doing it because we like it. So if you like what we do, please, you can go to GoFundMe to our campaign, and then just chip in a little bit, and then we can use this money to produce more cool events. And we're soon going back offline, hopefully. So we can really use this money. Also, if you got interested, you can join our next event. So now next Chit-Chat session is going to be on 13th of August. So Alexander is going to talk about dreams, so which is related to his research of memory and brain. So jump on and also ask your questions and enjoy it. Yeah, and so there is another thing. If you like networking, we have this this project, or this tool, you can call it, Random Coffee. So this is